it could prevent the virus from getting into the cell because it locks on the receptors. There's, different things. That's what B cells do. And the other two that we're going to talk about right now are the T cells. So we can say T cell or we can say T lymphocyte. They're T lymphocyte cells. It's the same thing. And there's three that are talked about in this module. So there's T helper. That's what we were just talking about. This is the best picture, so I'm keeping it on here, even though this is the cytotoxic T1. But we want to talk about both. So for the T helper one, it's similar to this because it is a T cell, but instead of CD8, it's called CD4. Right? So the T lymphocytes, both of them interact with cells. That's different from the B cells. The B cells don't interact much with other cells. They produce antibodies, and that's in your fluids. The T cells are cell mediated. It means they interact with cells. So the T helper interact with cells to get the whole immune system, charged up to fight an infection. The T cells do the opposite. They're cytotoxic. They go around and kill your own cells that are infected. So two totally opposite jobs, right? The T helper is the cheerleader, and then the cytotoxic T goes around and destroys your cells that are infected. But all the lymphocytes are specific. That's what's really good about them. So the B cells, the helper T, and then also the cytotoxic T, they are very specific. And so they interact through these specific receptors, right? They don't act on, they're not having these effects on the other cells of your body. They are having effects through these sort of specific interactions. So let's look at the cytotoxic T, and then we can sort of compare it. All right, so here's one of your cells and you guys can see this picture, right? The CD8 cell. Okay, so here's one of your cells that is infected. Maybe it's a virus. For some reason, it's infected. And then here is your T lymphocyte cell. And this is a cytotoxic T. Here they're abbreviating it TC. And its job is to kill your cell that is infected. And so what it does is it has a T cell receptor. And the T cell receptor is what binds to antigen. So I want you to know the receptors that you find on these cells. And so for a T lymphocyte cell, you're going to find this CD8, and you are going to find a TCR, which stands for T cell receptor. And so here you have your cell that's infected, and when it's infected, and the module goes into some detail about this, when a cell's infected, they start looking different on the outside. They might not make their normal MHC1 ID tags. So all of your cells have MHC1, and that is used to communicate and say to your immune system, hey, this is one of the cells. It belongs in the tissue. Don't attack it. Don't, it's a normal cell. This is how it can start learning the difference between infected cells or, pathogens or, and your regular cells. So is that in here? No. So it binds to MHC1 receptors or molecules on the surface of cells. So all your cells have that. If they are infected, then they will display on their MHC1 a little bit of that virus or that molecule that has infected them, and that very specifically communicates to the cytotoxic T cell that they are infected, and then they will destroy that cell. by making holes in it and then putting those digestive enzymes, and will destroy the cell. That's what cytotoxic T. In the module, it also talks about a related cell called natural killer cell. You don't have to know those as much. They're sort of kind of in between. They're not so specific as the cytotoxic T, but they are able also to identify when a cell is infected. It's just not as specific. So usually we consider that they're more part of your second line of defense. And these lymphocytes, the B and T helper and cytotoxic T, are third line. They're very specific. So questions about the immune cells or the receptors? Justin, anybody have any general questions about the T cells or the?
Yeah, I have a question.
Sure.
So the T cells, they only attack cells that are infected. They don't go look for other cells.
So you have different immune cells that can do different things, but the cytotoxic T are only activated when they bind to MHC1, and that is your self ID tag. All of your cells have that except for red blood cells. Red blood cells don't have a nucleus. They're different. That's partly why you can, get blood transfusions and it doesn't, your immune system is fine with it. But all your regular cells that have a nucleus, they all have MHC1. And it's just by random variations, we're all unique. And so unless you have an identical twin, you're going to be different from others. So your immune system can recognize your cells from something, from, other cells.
Okay, so if you have an identical twin, then like one of the twins' immune cells doesn't recognize its cells from like the brothers or sisters' cells?
No, they would, well, yeah. So the twins would be the same, they would be identical. But from siblings, depending on how, they'll check to see how similar, for if you're going to donate an organ or something like that. And so the more closely related, then it will be similar, but it won't be identical. So an identical twin would be an exact match, and your immune system wouldn't attack that tissue.
Okay.
Okay, perfect. All right. So let's see. Our next topic. We'll spend about. Class is fast, huh? Let's spend about 5 minutes on the staph and strep, and then we'll spend the last 5 minutes on the DNA fingerprinting. So let me find that. Let's see. Where is the staph and strep? Staph and strep. Okay. Okay, let me stop sharing for a second. Let me open it up and see if I can. Let's see. No, not that. Where did it go? Let's see. Actually, I don't think that's the right thing. I think it's the one before. Let's see. Yes. Was it week 6, I think?
It was, are you talking about the project?
Yeah.
Week 7, right? It was due. I don't know when you posted it.
Okay, yeah, I think it's in. Okay, so let me take a look. Let's take a look. Staph and strep. Okay. All right, so now let me share my screen again. Here it is. Okay. All right, can everybody see the staph and strep in here? All right. So the idea and what I want you to know for the exam is, so for this, you went through and you did this exercise, and it was also using exercise, you know, the staph and strep exercises in the lab manual. And so for some of you, it was exercise 32 and 33, for others, exercise 36 and 37. And using the results in this test, then you could use the table in the lab manual to identify a specific organism. So for the exam, I'm not going to ask you to identify a specific organism based on these results, but what I'm going to be looking for is that you understand what the tests are testing and what the results mean. So, so I'm not going to try and get you, you know, from the lab part of it. I'm not going to try and see if you can figure out which one is staph epidermidis or something like that. You may be able to, but I won't be asking it that way. I will be asking in terms of if you understand the testing. And so in identifying staph and strep, for instance, for gram staining, like know the characteristics. So for gram staining, they're both, you know, gram positive cocci. If we're talking about staph, they are catalase positive, strep, catalase negative. And you have to know what the catalase test is. And so I might have shown you before, but in addition to those lab exercises, you also can just look at the beginning of the lab manual, and it has most of these tests, maybe all these tests. I don't know, it might not have the antibiotic discs as a separate lab, but it has other ones. So for instance, here's the coagulase test on page 509. Or here's catalase 497. This is just in the index, or no, it's probably in the index, but this is a table of contents. And the reason that's useful is that it can remind you, and I would make a list of these tests. Okay, so it is hard because you're not actually physically doing them, so it's harder to remember them. But what you can do is, you know, I would make a list of all the tests that you did for staph and all the ones for strep in this. And so here's, you know, maybe even have like a little picture with it to help remind you. So it'll be quick when you get to that question on the exam. You won't have to be, you know, trying to find stuff. You'll just have it. And so you would say, okay, for catalase test, this differentiates between staph and strep. Staph are positive, strep are negative, and what's the test? Oh, it's an enzyme, and if the bacteria have it, then they will produce bubbles. It even has the reaction here, which I'm not going to ask you the reaction, but why is it bubbles? Because hydrogen peroxide is broken down and oxygen's released. Those are oxygen bubbles, and it happens right away. You add a few drops of the reagent on the bacteria. If it's catalase positive, it'll start bubbling. So I would do that for each of the tests that are in here so that you're more familiar with them. That's also arrangement. Staph are irregular clusters, strep chains. It could be a small chain. So any of the, you know, any of the media that are used in here, I would be familiar with it. Here's staph brom, or we say M. staph brom. This is specific for staph because it has a high salt concentration, about 7.5%. Most bacteria can't grow in that. So I think there was a question. There's different questions on the quizzes, so you might not all have gotten this question, but some of you got the question. If you're trying to isolate staph and you didn't have M-staph broth, if you use like a regular broth, like nutrient broth or trypticase soy broth, would that be fine to isolate staph? So staph would grow, but it wouldn't be a good choice because everything else would grow too. If you're trying to isolate staph, you want to separate them from all the thousands or could be millions of other bacteria that are in there. How are you going to find the staph? You're going to grow them on selective media. So this is media that's selective because it has salt. So know the difference between selective and differential. So selective means, and just write that down, just have your notes. And I think it actually talks about it in here, but selective, it has something in it to inhibit growth, like salt or dyes, or could be antibiotic. There could be something that inhibits growth of some, and so it selects in favor of others. And then another thing that media can be is differential. And differential lets things grow. It doesn't inhibit them, but they appear differently. Maybe there's a pH indicator, so the media turns color. It becomes yellow instead of pink. And you could have both. Mannitol salt agar is both. It has salt to inhibit, that's selective, and it has mannitol, which when bacteria ferment it, if they can, it produces acid, and then in combination with the pH indicator, it'll change the color of the media. So it'll look different if something is positive for mannitol fermentation compared to negative.
Can I ask a question?
Yes.
I felt like with this lab that you're going over right now, like we learned a lot about all the things that you're saying, like it being selective and kind of identifying them or what the results would be. But we didn't really go over like specific areas that you would find strep and staph. I had a lot of trouble with that on the quiz that was following this. So maybe we could talk about that in office hours. But basically, you're saying on the exam, something like that is not going to be on there, right? Like, would you find strep? What kind of strep would you find on fomites, for example? Something like that. It's not going to really be on the final.
No. Well, if you think it's not in the module and it's not in here and it's not in the lab manual, so it should be in one of those three places. But if you've checked those three places and you don't think that that was in there, send me a message and I will check. And if that's the case, I will regrade that question. So it should,
I may have missed it somewhere. I don't know, because
sometimes that happens. So if it didn't say that, I think in the module pages right before this, it talks about. It might not have talked about a fomite though. So take a look at those three, but if that's the case, send me a message and I will look at that.
Good.
Thank you for that. Okay. Okay. And so then finally, sort of narrows down and then using, so you won't need to do that for the exam, but I just want you to know the characteristics, know the tests. And so if I gave you a description, if I said, these are the results of this test, then I want you to be able to think about, does it seem like a staph? Does it seem like a strep? Which tests sort of support that or not? So be familiar with the test so that you can sort of analyze it and kind of interpret it. But I won't be asking you for names of species related to this. So there are species in the written part of the module. So this isn't talking about that. It's just as far as the lab question. Here's another one, coagulate.
They are my family. And me and Kelsey been knowing each other for a long time, since
so
like
these are just
2013.
So any questions about these tests? any of the tests that you're fuzzy about, or you just want me to
Damn.
clarify?
So for the multiple choice section, we're only going to be asked like if it's staph or strep based on certain test results.
That would be for this lab. Well,
For like-
what we're going over would be more like a longer type question, asking you to
He got in my mouth.
sort of interpret results.
What's up, GQ? It's
like
Megan Thee
if
Stallion, aka the Hot
it
Girl
turns
Coach,
pink
and
on mannitol,
it's definitely an item I cannot live without.
or
I mean,
if it forms a
how
clump and
y'all-
coagulates, or if it bubbles or doesn't bubble with the catalase test, being able to interpret that. I'm just saying that I won't ask you to get your lab manual out and figure out which species it is. I'm just wanting you to interpret the test. This is for lab. But separate in the module, there are different species like Strep pyogenes with different characteristics. That's sort of separate. And so you could still be asked about that, but that would be more like a multiple choice question. You know, what are the characteristics specifically of that species? But
Okay.
but for this lab, in the assignment, you had to figure out which species. For the exam, you don't have to do that.
Okay. That makes
Just-
sense.
Yeah.
Thank you.
Sure. Okay. And then the last thing is, let's just take a look at this DNA fingerprinting and polymorphism. Okay. So the main thing that that you should know is that polymorphisms, this is just, you know, most of our DNA is not coding. It doesn't code. It's not genes that make something like, you know, brain cells or hair or muscle or whatever it is. And for a lot of it, we don't know what it does. And then for some of it, we're learning, oh, you know, it actually does. You know, there are some of our DNA is for regulatory purposes and things like that. But 23andMe, Ancestry, these companies sort of use these variations that we have. And because polymorphisms are sequences of DNA that don't code for things, if they have a mutation, it gets conserved. So normally, let's say that my DNA just, you know, randomly mutated and it was in an essential gene, I probably wouldn't be able to survive, or that gene wouldn't get passed on, you know, to my daughter, and so on and so on. And so those changes, that's why you see changes in polymorphism that get kept for, you know, years and years and years and years, many generations, because it's not essential for the cell, so it doesn't stop the cell, you know, hurt the cell in any way. And so this can be used to tell the differences and sort of trace your kind of your ancestry, because it's known there are certain ones that they sort of figured out, you know, which part of the world they came from, and it's kind of cool. But with DNA fingerprinting, remember those restriction endonucleases, they're going to cut, and they will randomly cut the DNA at specific sites. And so it creates a fingerprint like your fingerprint in your finger that's unique because of the way that you can cut. And you wouldn't just cut with one enzyme. You would cut maybe, like in an extreme case, you could even go as many as, you know, with 13. And so you'll just get a very unique sort of profile for each person. And so that is how that's used, and it's just showing where it would cut. And then we could run it in gel electrophoresis by putting the sample of DNA after it was cut and then the little pieces will migrate. This is just showing a couple cuts, but if you imagine, you could do it like with your whole genome, and so it would just, be hundreds of or thousands of these pieces, and everybody's would be unique. So that's kind of the idea with that. All right, guys, I think we are over time. Does anybody have any quick sort of general questions before we
This is
end?
4 to 7, right? The exam is modules 4 to 7.
Yes, thank you. Yes, the exam is modules 4 to 7. It'll open on Friday and go through Sunday night. And message me, or you can come to office hours right after this class from 2 to 4, or on Friday night from 8 to 10. And if any of you, work and you can never do it from 2 to 4 or whatever, just let me know and we'll figure out a time that we could meet. So I do want you to be able to come and talk to me if those times won't work. Or you can just message me on the Canvas and I can answer questions that way too, if that's easier. Any final thoughts? Anybody?
Professor, so we have the exam 2, which is due by Sunday midnight, right? And also the quiz H is due at the same day. Is it like possible to extend the due date for quiz A because we have exam 2 this week?
Yeah, so for the only one that we don't, like for the exam, it's just those days. But for all the other assignments, you always have a 24-hour grace period. And so that one, if you're able to look at the quiz, it says that it's due Sunday, but it's like final due date is like Monday. So you do have 24 hours, so you could just do that one on Monday. the
Oh,
quiz.
thank you.
You can
I
always
didn't
do
know
that.
that. All right,
Yeah,
thank
you
you.
can always do that, yeah. Thank you for those questions. Anybody else? Okay. Happy studying. Write me if you have questions or come to office hours. Okay,
Thank
see you
you.
guys later.
Thank you.
Thanks.
Bye.
Have a good night. Bye-bye.
Thank you.
Okay, man. I'm just so tired.
Freaking clicking because we're all clicking the same one at the end. So
And I need to be calling y'all. Y'all need to be calling me. Definitely can't live without her.
You only have one phone.
I actually have like 4 phones, but this is the main one. And my other one, or actually my other 3 are dead all the time. I don't even know where they are right now, honestly. Like the other phones, I don't even know the phone numbers too. I've literally had my number since maybe the 6th grade. So everybody calls this phone all the time. These are